Hi students, I'm Dr. Anandi. Welcome to my channel Make Biology Simple. In today's video, we are going to see about the important questions expected for the biochemistry paper for the BDS students. So we'll move on to the important questions and the repeated question asked in the exam. So the first and the foremost questions will be asked in the biochemistry chapters are one is the classification of lipids. So what is classification? Classification of fatty acids and describe the reaction of beta oxidation of palmitic acid and how many ATPs are generated and complete oxidation of 1 mole of palmitic acid. So you can see that in a single question, three subtypes of questions are asked. So classification of fatty acid, you have to write simple lipids, derived lipids and complex lipids. And coming to the reaction of beta oxidation of fatty acid or palmitic acid, both are same. So palmitic acid specifically is asked, so see it is C16. So C16 means it is converted to a two carbon form called acetyl CoA. So 7 cycles it repeats and exhausts this palmitic acid. And how many ATPs are captured when oxidation is? You have to write it as you have to write as 129 ATPs are generated in the thing. And describe the second question is outline the steps of glycolysis and how it is regulated. Add a note on energetics of glycolysis. So again, glycolysis is nothing but is glucose is converted to pyruvate. So that is called the glycolysis, and how it is regulated with the help of the feedback regulation and some of the product as well as the concentration of the substrate and availability of the molecules which is required for the running of the glycolysis. All these you have to write. These are the points to be included in the glycolysis. And then add an energetic means for one molecule of glucose is oxidized. You're getting 8 ATPs in the case of aerobic. As in the case of anaerobic, only 2 ATPs are generated. So this is the answer you have to write for this. So next thing is again the steps beta oxidation of palmitic acid is asked and give the energetics. So I think I repeated the first question. The same thing you have to write it for here also. And the fourth question is about describe the using the graphs factors affecting the enzyme catalyzed reaction and write briefly on the competitive inhibition of an enzyme. So what is asked is graph. So factors affecting enzyme catalyzed reaction, we have lot of things. 7 factors will be there. A substrate concentration, effect of temperature, effect of pH, effect of product formation, and all these are coming under activation energy. So for all the factors, there is a graph which is explain the enzyme catalyzed reaction. So you have to write the factors and the graph also we have to include for this question. And competitive inhibition means out of three inhibitions, you have to write only competitive inhibition. So that is the one which is required for this question. And coming to the sixth question, you can see that tricarboxylic acid cycle. So tricarboxylic acid cycle is nothing but TCA cycle or citric acid cycle, or we call it as Krebs cycle. So all are together same only. So you have, if any way it is has, you have to write only this. So what is TCA cycle and this? And write the energetics and its significance. So energetics again here it will be produced not directly ATP. Instead of that, they produce the electron carriers like NAD and FAD and ATP. That is nothing but GTP. Here it is produced. So 1 NAD is equivalent to 3 ATPs. So like that you have to calculate and express the ATP productions. And in the case of FAD, it is 2 ATP. So GTP is equivalent to 1 ATP. So this is the calculation how we have to proceed for the energetics. And next thing is hormonal regulation of the blood glucose. Hormonal regulation means you have to write all the insulin. Insulin is the hormone which decrease the blood glucose level. And they said there is an antagonist hormones called glucagon, epinephrine, and thyroid. So all these are coming under the antagonist of it. All these are going to decrease the glucose level. Sorry, this only in decrease, and here all will increase the glucose level. So these are the set of hormones which will regulate the glucose concentration. So this is what you have to write for the this question. And the next question is this asked about the reactions. Reaction involves the de novo synthesis of palmitic acids. So there is always a confusion among the students. There is a difference between beta oxidation and de novo. De novo means synthesis. Beta oxidation means degradation. So palmitic acid means again it is E16, but it has to produce from the acetyl CoA. So acetyl-CoA will combine with a carbon dioxide and form malonyl-CoA. Like that, it extends and produce malonyl-CoA, that is 3 carbon, and it is extended to produce the formic acid, which is C16 carbon. Formic acid. So like that, it gets on extended. And what is the coenzyme required here? The CoA enzyme is nothing but NADPH, because we need a hydrogen also for the extension of hydrogen group. For example, you know that the formula for any fatty acid is CH2N times and then COOH. So this N times of hydrogen has to be extended with the help of the NAD. So NADPH will donate the hydrogen. So you have to write that alone for this. And this is you have to explain the steps. And again, the seventh question is beta oxidation of saturated fatty acid and energetics. So as I already repeated the same question in the earlier slide, same C16 palmitic acid is converted to acetyl-CoA and 129 amino, ATPs will be generated in the bioenergetics of this. And a formation of ketone bodies. So ketone bodies is nothing but acetone formation, acetone and acetoacetate, acetate, and finally beta hydroxybutyrate. So all these are nothing but ketone bodies. So these are again depend upon the from the fatty acid, that is acetyl-CoA is the one which required for this also. So these are the points to be covered for this question. And then glycolytic pathway, just now I gave same thing is repeated. So most of the repeated questions only I am discussing now. And then what is a normal plasma glucose level? So you know that it should be 80 to 120, and this is the level of fasting condition, and explain the mechanism involved in the hemostasis. So hemostasis means as the blood glucose increases, it is all stored in the form of glycogen. As the blood glucose level decreases, so from the glycogen it is getting back to produce the glucose. So this is a step how glycogenesis, glycogenolysis. So this is what the mechanism involved for the glucose hemostasis. Hemostasis means maintaining the glucose concentration in the plasma. So you have to write genesis, genolysis pathway and the normal concentration. And then the 10th question is about the 10th question is about the essay on the resources of RDA, biochemical function and deficiency symptoms of vitamin D. So whenever we are studying for the minerals and vitamins, you have to concentrate on this. So write the sources which is rich in vitamin D and how much is required for the adult and the children and the elder people, and what is the main biochemical function of vitamin D, that you have to write, and then deficiency. So these are the direct points you have to study and write it for this question. And next question is glycogenesis. Glycogenesis is nothing but synthesis of glycogen. So that is nothing but glycogenesis. So as I told you, whenever the glucose concentration is increased, it proceeds to the pathway of glycogen synthesis. That glycogen synthesis is nothing but glycogenesis. And add a note on glycogen storage disease. So whenever the enzymes are deficient, that inherited disorders all will be there. So as a result, you have this glycogen storage disease. You have to write the set of storage diseases for this question. And then write the metabolism of glycogen and its metabolic defects. So same thing is repeated here. Metabolism of glycogen means not only genesis, you have to write genolysis also, because this pathway is initiated by synthase and degradation started by the phosphorylase. So the enzymes and the hormones are regulated and it proceeds either genesis or genolysis depending upon the glucose concentration. So particular phosphorylase or the glycogen synthase defect enzyme is there, that leads to the metabolic defect. So that is what I have explained here. Okay, next, detailed various biochemical reaction involved in the beta oxidation of fatty acids. Beta oxidation of fatty acids are nothing but they are involved in the again the same question is repeated here. And the next question is describe urea cycle. So urea cycle is, there is a specific pathway for this. So you have to write the urea cycle and mention the normal blood urea and causes of uremia. Direct questions, you read as such and then write it again. So the next question is about what are the dietary sources of RDA and intestinal absorption of vitamin D. This is also repeated. And beta oxidation of fatty acids and note on energetics is repeated. And how blood glucose is regulated is also repeated. The dietary requirement, daily requirement, absorption, Absorption and functions of calcium. So this is new question. This is coming under minerals, macro minerals. So you have to write what is the RDA and what are the sources, and absorption, because absorption is favored by the parathyroid hormone and vitamin D. And then functions of calcium you have to write. So this is also direct questions. You can write it from the exam point of view. You have to write all the points up. And this is also repeated intervals of fasting plasma glucose hemostasis. That's just now I explained what to write. And then beta oxidation of fatty acid, mention of see, this is again and again repeating, and this is also repeating. Normal postprandial level means after the intake of food 2.5 hours, what is the level? So it can goes up to 180 milligrams. So like that, this is the what you have to write, and read in detail of regulation of blood glucose. Detail is about, again you have to write the hormones. Hormones is the one which going to control the blood glucose level in either in all these conditions, excess intake of glucose or less intake of glucose. So all this you have to study. So this is what you have to write for this question also. So this gives about what are the questions expected from the carbohydrate unit. So in the carbohydrate assay, you can expect this glycolysis pathway, TCA pathway, and gluconeogenesis. So gluconeogenesis is different because glucose synthesis from the non-carbohydrate compounds is nothing but gluconeo. Neo means non-carbohydrate compound. Genesis means synthesis. So you have to write from the non-carbohydrate compounds like amino acids, then fatty acids, then glycerol. So from these also you can able to produce a glucose. So these are the points which have to be included in the particularly for this question. Then hormone blood glucose level regulation, we know that glycogen metabolism. So this is what earlier I repeated in the questions. And coming to the lipid chapter, these are the unit questions. Beta oxidation and ketone bodies metabolism. Along with that, one question is expected as a synthesis. That's also you can write. And coming to protein assay, you can essay questions. You can go for urea cycle. This is also commonly repeated questions. So coming to enzyme chapter, classification of enzyme, you have to write this OTHLIL. OTHLIL is the one which you have to remember easily. O for oxidoreductase, T for transferase, H for hydrolase, L for ligase, I for isomerase, L for lyase. So this is the one which for each thing you have to give one example and the nomenclature. So if you write all this, that is enough for this question. And factors affecting is also repeated. Enzyme inhibition, competitive, non-competitive, irreversible, all this you have to write. And then clinical diagnostic importance of enzymes. So there are either you can ask like this or allosteric enzymes. For example, LDH. For example, LDH. For example, LDH, you can write LDH, creatine kinase, alkaline phosphatase. So, set of enzymes are there. So, you can write according to the essay questions. So, coming to biological oxidation chapter, electron transfer chain is asked, or mechanism of biological. Both are same only. So, it is asked in different form. What is electron transfer chain or mechanism of biological oxidation? It takes complex 1, complex 2. So, it is involved 1, 2, 3 is there. 4 is there, 5 is there. So 5 complex is a 1 ATP is generated. So this is how it has been produced. And coming to nutrition and mineral point of view, vitamin A, vitamin D and calcium is required, is expected. Apart from that, you can also study iron. Iron is also a very important question, and C is also sometimes asked. So these are the important questions expected from the each chapters. So next we are moving to the short questions. So what are short questions? Short questions is yeah, you will be asked a set of 4 questions. So 4 into 5, 20 questions will be there. So first and the foremost is urea cycle. What is expected in the essay is also asked here. And classification of enzyme with examples, structure of DNA, coenzymes of niacin. So for structure of DNA, you can have to write a double helix, and then how many bases are present in the major and how many the bases present in the minor helix. All this you have to explain. And this is nothing but Watson and Crick model. It's nothing but Watson and Crick model. If you write, that is enough. And then coenzymes of niacin, it's nothing but NAD and NADP. So these are the two coenzymes which is derived from this vitamin B, niacin. So next thing is the next set, these are the questions is collected from the question bank. So each year will be vary. So other set of questions you are expected is formation and function of calcitriol. So calcitriol is a hormone. You can expect, you can write the, like this. What are the main functions of calcitriol and how it is formed? That means synthesis. So that is enough because it's a short notes. No, you can write it simply. And absorption of iron. I told you, no, iron is another mineral which is expected. You can write this. You can study iron also. Again, structure of DNA is asked. Isoenzymes is also repeated. Isoenzymes, as I told you, LDH, creatine kinase, alkaline phosphatase, and then amylase. So these are the expected enzymes for the isoenzymes. So other set of questions in the 1 year is they are asking about oral glucose tolerance test. That means you have to consume a glucose at particular concentration, and every half an hour you have to take the blood and you have to note the concentration. So that is nothing but oral glucose tolerance test, or called as GTT. So you have to write and you have to put the graph, and you have to show how much of it should be increased from 80 to 180, and then it should be decreased and coming back to 80. So this is the graph you have to explain. And absorption and function of calcium is very important. So this you can study it for essay as well as for the short questions. And immunoglobulins, this is IG. So IG is types, so we call it as GAMED, GAMED. IgG, IgA, IgM, IgD, and from the IgG you have to write the structure like this, light chain, heavy chain, and then how it is interpreted. All this you have to write, and what is the NH2 terminal contains, what is the COO terminal contain, all this information you have to write for the immunoglobulins. And phenylketonuria is you know that it is a aromatic disorders, aromatic amino acid disorder, so that that also you can write it under the phenylketonuria. So the next set of questions is dietary activation and function of vitamin D, that is same, and BMR, basic metabolic rate, and what is the clinical significance. You can assess the kidney function, you can assess the liver function, all this you can study, thyroid function, everything you can study for the clinical significance by measuring the BMR. Pentose phosphate pathway is one of the important pathway in the carbohydrate metabolism. It is nothing but pentose phosphate or hexose monos phosphate pathway. So you have to write the steps starting from glucose 6 phosphate. It ends with the glucose 6 phosphate. So both are same. So that's why it is a this is a peculiar pathway, but in between it produces a ribose, which ribose is very important for the synthesis of DNA, nucleic acids, RNA. And then it also produces NADPH. NADPH, you know that it come across in the synthesis of fatty acid. So fatty acid synthesis, we need this because the hydrogen is donated for the carbon extension. Hydrogen is coming from air that is generated in the HMP pathway. So that is enough. And metabolic defects in the phenylalanine metabolism. So phenylalanine metabolism, you have to write this, all this phenylketonuria, all this will have to come. These steps you have to write it. And other set of questions are immunoglobulins, just now I explained, sources, requirement, and functions. So here you can see that other set of questions are dietary sources, biochemical functions, and manifestation of vitamin D, and then transamination. Transamination is nothing but a set of reactions where it carries a keto and amino group. A set of keto acid and set of amino acids. So together is called transamination. Galactosemia is nothing but the galactose concentration is increased in the blood. So this is called galactosemia. The metabolic disorder you have to write for this question, and then metabolic defects in the metabolism of glycolygen. So this is also a repeated question. So next set of questions you can expect BMR. So BMR is basal metabolic rate, and what are the factors affecting this thing is very simple and easy question. You can attend and score more marks for this. And again, normal serum calcium level, and what is the role of vitamin D, because without vitamin D, serum calcium level, it cannot be what to say absorbed. Even though we consume calcium, it is not absorbed because we need vitamin D for the absorption. So what is the normal serum calcium level? You have to write that is 9 to 11 milligram per deciliter. That you have to write specifically. And fluoride is very important for the proper coloring of the teeth and enamel formation. So all this have to write for the functional and physiological importance. And significance of pentose pathway, I repeated, production of ribose is produced as well as NADP is generated. So this thing are produced in the HMP pathway. So that is the significance of the HMP pathway. So that you have to write. And the other set of questions are lipoproteins. Lipoproteins, you have to write chylomicrons, VLDL, LDL, HDL. All this you have to write, and this importance for the carrier of the lipids in the circulation. Blood buffers you have to write in detail. Bicarbonate, phosphate buffer. All this you have to write. Plasma proteins. There is a chart for this. You have to write all the albumin, globulin. Under globulin, what are the subtypes you have to write, and then fibrinogen. So these are the set of proteins which is coming under the plasma protein. Ketone bodies, as I said, it have a acetone, acetoacetate, and beta hydroxybutyrate. This you have to write. So here, next thing is sources. Again, you have asked sources of vitamin C, and classify lipids is given. We have to write simple, derived and complex with the suitable examples. And hormonal regulation of calcium, we have parathyroid hormone and calcitonin. So all this you have to write for this. With the metabolic pathway, and then essential amino acid and their function in the body. Essential amino acid you have, so you can write a set of things you can see that. So you can easily remember this. Methionine, isoleucine, lysine, leucine, phenylalanine, histidine, tyrosine, tryptophan, valine. So you can remember this and can write the functions of the body. Function means any protein depend upon these amino acids. So without this, the protein synthesis is not complete. So you have to write the synthesis of any protein requires the essential amino acids. Another set of reactions you can see: lipoproteins, fluorides, gout. So gout is the degradation of the degradation or deprivation. What to say? What to say? Degradation of nucleic acids. That means that is very important. That is purine degradation. So that is what from purine only gout is produced. That means uric acid. Uric acid is, if it is increased only, we call it as gout. So it is a disorder of purine metabolism. Very simple. And again, enzyme classification with the examples. As I told you, OTHL you write, and with the example you have to write all this. And other set of questions are hyaluronic acid, protein denaturation. Protein denaturation is is one of the factors which is coming under the proteins. Hyaluronic acid is very important in the mucopolysaccharides and polysaccharides coming. Obstructive jaundice is because of the obstructions in the liver and the pancreas. You can expect this jaundice. And creatine clearance test is a kidney function test. You can write this exactly. So it's all straight questions that to the point you can write and score more marks. And the last set of question, again you have to write what is mutarotation, essential fatty acid, structure of DNA, kwashiorkor. So kwashiorkor is nothing but protein energy malnutrition. So because of this protein energy malnutrition, it's affect the children's. So mutarotation is nothing but the glucose or the carbohydrates which is dissolved in a solution, and the specific rotation it is nothing but we call it as levodextrorotatory. All this points should be covered in the mutarotation. Essential fatty acids is nothing but all the polyunsaturated fatty acids, say for example linoleic, linolenic, arachidonic and DHA, all this will come under essential fatty acids. And then again fast examples with the enzymes classification, and then normal fasting and glucose level, urea cycle, calcium. This is all repeated questions. And the other set is function of iron, and here specifically they ask vitamin A. So we call it as wall cycle, no? Wall cycle. You have to write the wall cycle for this question. Substrate level phosphorylation is during the metabolism itself when without entering electron transfer chain, if in the pathway itself ATP is produced, that is called substrate level phosphorylation. Then vitamins and antioxidants. There are specific vitamins, say for example A, vitamin C, vitamin K. These are all acting as an antioxidants. That nothing but it is scavenging the free radical formation. So that's why it is called vitamins with a specific activity with antioxidants. And the next set is, again you have seen that structure of DNA, scurvy. Scurvy is deficiency of vitamin C. RNA polymerase is involved in the synthesis of RNA. Kwashiorkor is a malnourished disorders. And then phenylketonuria. Phenylketonuria is disorders of the aromatic amino acid metabolism. Function of calcium directly you have to write. The formation of bone and teeth, all this you have to write. Irreversible steps, you can see out of 10 steps, only 3 steps are irreversible. You write these 3 steps alone, that is enough. Dietary fiber is along with the nutrition part, you have to write dietary fiber, what is its sources, how it is involved for the metabolism. That is enough. And the last set of question is beta oxidation of fatty acids. It's already asked in essay question, here also in right. Chemoosmotic theory is a theory which explains how ATP is produced because of the energy transfer, the proton transfers, push of protons from the IM space to the matrix. So that is one which helps for the ATP formation. So that is simply I'm telling you what is chemoosmotic theory. Then obstructive jaundice, again repeated, respiratory acidosis is a disorders of the acidic form. Now, all these are very important, simple questions only. So I think you can answer all these questions. That will be very useful for you. I think this slides and this video will give a clear idea how we can prepare for your exam and how we can write for your exam. What are the points to be covered during your final exam? So I hope this video will be very useful for you. Thanks for watching. Thank you, students.